Everybody to the next session. My name is Tima Komludi and I will be your chair with Professor Werner Koopman. And our next session will be about the perspectives of bioenergetics in health and disease.
Yes, welcome everyone. So let me introduce our first speaker, which is Dr. Lubos Sobotka, and he will talk with us on the Crabtree effect in nutrition. Thank you.
Okay. First of all, I would like to thank for kind invitation to this conference. I am clinician, internist. Therefore, I would like to apologize to people who will feel that I am telling two basic things. Topic of my talk is Crabtree effect and clinical nutrition, and it will be mainly focused to carbohydrates or glucose, because present point of view for sugars in diet is that carbohydrates are mainly source of energy, or if they are only source of energy, and that we should give not more than amount of carbohydrates that is oxidized. So our point of view is that our body is working like steam machine or steam engine. It burns carbohydrates and use it only for energy. Energy in mitochondria, and it produces ATP. And optimally, it can produce 38 molecules of ATP. Under anaerobic conditions, 2 molecules of ATP. And of course, the more is better, which means that aerobic metabolism is better than anaerobic metabolism because it produces more energy and more ATP. This is general point of view of clinicians throughout the world. I would like to say that glucose is the most abundant molecule, or glucose polymers like cellulose and glycogen are most starch are most abundant molecules, organic molecules, because they are synthesized, everybody knows, from CO2 and water. And glucose molecule and its metabolites are basis for all other molecules. Yesterday we have seen about reverse Krebs cycles, and I would like to stress there is many reverse cycles in the nature. This is one of them. It's regeneration part of Calvin cycle, which produces ribose 5-phosphate for Rubisco. And we have pentose phosphate cycle, and non-oxidative part of pentose cycle is reverse of Calvin cycle. So Calvin cycle utilize hydrogen, pentose cycle produce hydrogen or produce NADPH. And this NADPH is very important for many metabolic purposes, like reductive biosynthesis, because biosynthesis is mainly reduction, oxidative defense, also detoxification, signal transfer, immune response. And the main source for this NADPH is glucose or glucose metabolized. Glucose in pentose cycles and malate and other substrates from glucose metabolism. What does it mean, Crabtree effect? Crabtree effects are two effects. One is described in yeast. It was described by Crabtree, and it's production of ethanol when there is abundance of glucose. Then yeast stop oxidize glucose and they produce ethanol. In animal cells, it's Crabtree effect in tumor cells, which is also known as Warburg effect. And also it's and Crabtree described that this effect is also in dividing non-cancer cells. Our data, in vitro data, we demonstrated Crabtree effect in hepatoma cells, where we showed then during routine oxidation, high glucose medium, in high glucose, the oxygen consumption was lower than during low glucose, and when we added glucose, we decreased routine oxidation, And no change, of course, was when the cells were growing on high glucose. Clinical studies. There are clinical studies which were not explained yet. This is one of them. Ole Lundqvist described that if they gave carbohydrates before operation, insulin resistance was better. He has no explanation. When I was talking to him, he told me that it's probably run metabolism or something like that, something diffuse explanation. So we did some studies in our department, this is one of them, where we show that fasting in diabetic patients decreased glucose tolerance, so it increased insulin resistance. It's euglycemic clamp. But you can see that non-oxidative metabolism of glucose was not changed. It only changed glucose oxidation, decreased glucose oxidation. Another study which we did was study in critical ill diabetic patients, and we showed that we kept plasma glucose level at about 9, but we had two groups of patients. One received high amount of glucose, 6 grams per kilo per day, and second group received half of that together with fat. What we found was that insulin infusion was the same, which means that glucose intake improved insulin resistance. And this is our recent study with type 2 diabetic patients which received extra carbohydrates in form of maltodextrin after each main meal. And this is day 1, 2, 3, and this is day 4, 5, 6. And what you can see is that after they received some significant amount of glucose, their insulin resistance was lower than before glucose application. Do we have any explanation? We can have explanations that glucose is important building block, and we have very small amount of glucose or carbohydrates in our body, which means that we must have insulin resistance whenever glucose amount is low for non-energetic purposes. This is slides which we published with Andre, and we showed that this is turnover of glucose lactate and increased production of lactate during all conditions when we need more substrates for all these purposes. So non-oxidative glucose is important for synthetic, anabolic, and defense processes. And glucose is much more than only energy substrates, but it's more universal substrates. Thank you for your attention.
Thank you. There's time for some questions. Over there. Who gets there first? Okay.
To see if that was sort of the primary mechanism that explained why the glucose per se was improving its own uptake in an insulin-independent way?
No, we only combined hyperinsulinemic clamp with indirect calorimetry, and we measured this.
Thank you.
How do you explain the resistance of clinicians to listen to you?
You know, I think that we are 10 persons probably in the world who tell this. But I don't know how, you know, because these are our results, you know, but they say that glucose is toxic. You know, it's almost prohibited to eat carbohydrates for children. I don't know. I think that even obesity is not connected with carbohydrates. Obesity is connected with fat intake, because it's not so easy to be fatty with carbohydrates.
Well, to follow up on my provocative question, your studies are on patients that are quite immobile, is that correct? I mean, are there any studies on, say, highly or normally exercising persons? How then the
I think there are, I think everybody who deals with sport medicine knows that to have muscles, you need basic amount of lipids. They don't eat more than some basic amount. Not too much proteins. They eat 1.5, 1.8 grams of proteins. Am I right? I think there are sport medicine boys here. But they eat a lot of carbohydrates during and after exercise. And without carbohydrates, they have Rett syndrome. Therefore, if you want to gain the muscles, I think we should learn from sports people because, sport nutrition is connected with money. So what we can learn is from sport people and from meat-producing companies, because but I don't understand it, because if you exercise and they do it, they don't eat fat to have muscles. They eat up to 400. By cycling, people eat up to 1 kilo of carbohydrates per day.
Yeah, also thank you very much for the nice presentation. I would like to ask, maybe it's not directly related to what you observed, but maybe you could state about the potential role of chronic low-grade inflammation that you also see in terms of diabetes. So have you considered the concept how inflammatory processes might mediate the way the cells produce energy in the context of diabetes?
Yes, it's known that aerobic glycolysis is not only typical in cancer cells. It's not in cancer cells. It's in cancer, because cancer is not cancer cell. It's not one cell. It's whole. It's known that during inflammation we produce much more lactate and we have high glucose turnover. And it's produced by inflammation because we must, inflammation is in general catabolic process, but locally it's anabolic process because we have anabolism in bone marrow, in bone marrow, we are producing more inflammatory cells and so on. Therefore, we need substrates for these anabolic processes. And what we do is that we decrease, and it's demonstrated as insulin resistance, because insulin resistance is typical for inflammatory process.
So if I'm allowed, I would like to ask a second question. So do you also, or would you also expect to see dose-dependent effects in the change of the metabolism, considering, for example, body mass index or body mass excess? If I remember the discussions with Erich about inflammatory processes and the potential coupling mechanism on cell metabolism?
First, as Eric told, at this time, I am happy that I was allowed to do these clinical studies, because, but I have, from this aspect, I have the study which we did, the study in ICU patients, but it was a group of 50 patients. With 50 patients, it's very difficult in ICU to differentiate muscle growth and so on, because we only know that glucose resistance was probably better, because they have the same amount of insulin. Of course, the basic idea, which was my clinical experience, I know that patients who start to receive more glucose are better, but it's my clinical experience, which is, it's not bad because I have long time clinical experience, but But it's just, and therefore now we started to do some studies, but it's extremely difficult to have cooperators.
Yeah, perhaps go to I think there's a question in the middle still. Short question and short answer, please.
Short question. There's a seminal paper by the Weizmann Institute in which they followed 800 people and showed an immense variation in how the individual response is to nutrients. So one person gets into hypoglycemia with a banana and not with a cookie, and the other person just around. Was there also variation in your patients, or are they so stressed that it's overruled?
Yes, yes. It was, of course, there were some patients who respond by higher increase of glycemia, especially during first days, but and there were patients who profited immediately after glucose. But in general, the trend was that glucose intake increased, decreased insulin resistance in type 2 diabetic patients.
Okay, thank you. I guess we need to move on to the next speaker. Thank you. The next speaker is Dr. Moreno-Sanchez, and he will discuss with us let me check what he will discuss with us, the Warburg effect in cancer cells. Thank you.
Hello, thank you. Before I forget, I would like to thank Eric and the Ouroboros team for the opportunity to present my data and to be here. Okay. Yeah. Otto Warburg in 1956 published in English, this statement: all cancer cells are glycolytic. Since then, everybody believed that. Many people don't even assess this experimentally. So what we decide to do is in all of our cancer cell lines and models to determine experimentally the glycolytic fluxes. This is just a small sample. Okay, let me see. No. The first lane is with HeLa cells. You probably don't know if you are not in the field. HeLa cells don't have p53 transcriptional factor. It's a very important transcriptional factor, controls gene expression of many different processes. So it's a big deal in the cancer biology field. These cells don't have p53. The second one is the same HeLa cells, but push overexpression of wild-type p53. The third line is HeLa cells with p53 with a mutated p53, the most common mutation, which is claimed is a gain-of-function mutation. And the other ones are human breast cancer cells. This lane is from MCF7, is a non-metastatic cancer cells. The other two are highly metastatic cancer cells. People in different laboratories use this other model, as a cancer cell model, but probably it is not. As comparison, or for control experiments, we use this fibroblast as control. And then I'm going to discuss and show you some data with this tridimensional model. These other ones are bidimensional models, traditional models in Petri dishes. You culture the cells and you measure things. But the most close model to a solid tumor is a tridimensional model, what we call microspheroids. You can see here lactic production. We think that's the most appropriate way, to measure glycolysis. No people, when they decide to measure glycolysis, they measure glucose uptake. or glucose consumption, and they call it glycolysis. Or, lately, acidification rates. We think the most appropriate way to measure glycolysis is measuring lactate production. But lactate can also come from glutamine oxidation. So let's not forget that. We have to correct then for lactate production. In the second column, we correct for glycolysis by using 2-deoxyglucose, a glucose analog, which inhibits glucose transporters, exokinase, and exophosphate isomerase. Let's not forget that. It's not a specific inhibitor, inhibits several enzymes, but blocks the upper part of glycolysis. You have to correct for that. And after that, we use rotenone to correct for and get glutaminolysis, the last column. Okay, in the middle column, you can see the high glycolytic rates for all these cancer cell models. Very high glycolysis fluxes. So, at least in this part, Barbour was right, at least in our models. Cancer cells are glycolytic. Okay. The microspheroid, I wanted to show you, this is real art in our group. There are few people can do this. This is 5 days. After 2 weeks, that's what we call young microspheroids. There is no oxygen or carbon source limitation, diffusion. All of them are proliferating. But in the mature, after 3 weeks, in mature microspheroids, there are oxygen limitation, carbon source limitation, diffusion, and you get here a model for solid tumors. You have the proliferating cell layers. In the middle, you have the not so proliferating cells, and in the center, you have an apoptotic necrotic core, like in solid tumors. Okay. This is the publication in 1956, but the second part of what is called now the Barbour hypothesis, Barbour effect, is this other one, which people tend to forget. This was the second part of Barbour hypothesis. Respiration of all cancer cells is damaged. He said mitochondria are impaired in cancer cells. 10 years later, in England, in this meeting of the Nobels, he went further and said, when mitochondria are damaged, that's the prime cause of cancer. Let's not forget this. This is the Barbour hypothesis. Me being a mitochondriologist for so many years, we decided to go and measure oxygen consumption rates in our cancer cell models and oxidative phosphorylation. Okay. So we evaluate experimentally the second part of the Barbour hypothesis with this simple protocol. This is the oxygen concentration with cells, and then they start consuming oxygen. After a while, we add this particular, very specific, potent, permeable inhibitor of ATP synthase oligomycin. That's our simple protocol. This rate, routine respiration, minus this other rate, leak respiration. We also call it oligomycin-sensitive respiration. We call it net OXPHOS flux. I agree with Eric. We can call it this way too. Okay. We measure then in different cancer cell lines the same cancer cell lines, all the three, HeLa, wild type, HeLa with wild type P53 overexpressed, and the mutated one. Oxygen consumption, the routine respiration has to be also corrected, particularly for cancer cells. Let's not forget that cancer cells overexpress other enzymes outside of mitochondria, particularly P for 50 cytochromes, which consume oxygen. Let's not forget that. We have to correct then, for oxygen consumption rates with oligomycin. So in this column, we call it net OXPHOX fluxes. You can see the values. All of them are significant, are high. All of them, all these models, consume oxygen. All of these models have functional mitochondria, all of them. Well, with this data, before that, I wanted to show you, these are the original data published in German in 1923, 100 years ago. This is the original data, and here is the data. Liver has these oxygen consumption values, but liver carcinoma, surprisingly, about the same. But he went on and on saying mitochondria are damaged. Okay, well, you probably want to know this. This has become the cancer metabolic central dogma. Everybody believes that glycolysis is the predominant energy metabolism pathway. This has to be updated, but at least these three groups believe that. They don't measure it, but they believe that. Okay, with our glycolytic fluxes and OXPHOX fluxes, we determine the ATP synthesis by using, for oxygen consumption, the PO ratios. These are the values here. So except for this one, HeLa cells with mutated P53 and microspheroids, the mature microspheroids, here other one, all other cancer cell models, the ATP supply is provided by oxidative phosphorylation, not by glycolysis. Okay. We use this knowledge to titrate two properties of metastasis. These are very important properties. One is invasion. The metastatic cells invade tissues. We measure invasion. And migration, they move. We titrate with oligomycin OXPHOX, and you can see here, you can get 50% lower oxidative phosphorylation capacities and no great effect on invasion. But what about migration? Complete dependence. Migration is completely dependent on oxidative phosphorylation. Okay, last one. Our conclusions, oxidative phosphorylation is the main provider of ATP for many cancer cells. No, there are exceptions. And OXPHOX can be targeted to fight cancer. Thank you.
Thank you. We have time for one or two questions. Two, one in the middle and one
very nice, Rafael. I just want to mention that I agree with what you said, and but I would say that cancer cells don't need OXPHOX, and for example, rho-zero cells can make ATP very efficiently using glycolysis. So I think that cancer cells are highly plastic. Their plasticity allows them to use whatever energy is available given the conditions they are in. So that's all I want to say. Thank you.
Okay.
Very interesting presentation. Lactate is also considered an oncogene, and I think if you block or ablate LDH, actually the progression of cancer cells is reduced. And so is oxidative phosphorylation protective in that realm? Because again, mitochondria can oxidize the lactate carbons.
Okay, as a substrate, a carbon source, yes, lactate can be taken up by cancer cells, yes.
Yeah, so maybe it's a protective effect rather than a detrimental effect to have.
At what concentrations? Over 10 millimolar or higher. Not at lower concentrations. At physiological concentrations, no. Probably at higher, yes, like 40%. They publish with 40% these other effects, yes. Okay.
Yes, one final question in the middle. Sorry, there's no more time for another one. Sorry. Yeah, please.
Thank you for the great talk. So I tend to agree with you in the sense that it makes sense why mitochondria would play such a central role, given that we know that they're excellent at sustaining survival in mammalian cells. One question I would ask, and that I think about a lot, is to what degree do you think that oxphos is actually serving the purpose to provide sort of reserve for endergonic processes like glycolysis versus being used to fuel biosynthetic processes?
I think I'm not able to answer that, but we don't have access to metabolomic instrumentation, not there. I try to look at that in Rio, but okay, well, Lula, Vilma, and Bolsonaro, they think, well, okay. We haven't measured the anabolic role of mitochondria in cancer cells, no. I agree with Jiri. Glycolysis is also required for both glycolysis and oxidative phosphorylation, but glycolysis' main role is anabolic role, to provide precursors, no ATP. Yeah, okay, that's my answer.
Okay, thank you. We now will move on to the third speaker of this session, which is Dr. Porter, and he will show us a bioenergetic comparison of different cancer types. Thank you.
Okay, first of all, I'd like to thank Eric and the organizers for inviting me. I don't take these things for granted, so it's always a pleasure to be here. And as usual, the hospitality and the exertion levels are always very high. So I'm going to talk to you today about a subset of work that was part of a PhD project for a student of mine called Marilena. And I'm going to talk about comparison of bioenergetics in human tongue, precancerous dysplastic oral keratinocytes and oral squamous cancer cells. And that then was part of a bigger project on cancer phenotyping these cells. And that then was part of a big grant on oral cancers funded by the Marie Curie to 11 principal investigators. And Eric was involved in this as well. So in this space, there's not much information. So it's virgin territory. So anyways, let me give you some background. So oral squamous cancer cells are the 6 most common causes of cancer. They represent 90% of oral malignancies. They occur on the lips and the tongue, and we looked at the tongue cancers and mouth flora. The protagonists in causing them are tobacco, excessive alcohol drinking, which we better mind ourselves about, and human papillomavirus. And then they're usually caught quite late, like dentists usually diagnose them as opposed to doctors. But the treatment is quite severe: surgery, radiation, chemotherapy. There's no drugs in this space, really. And as I said, diagnosis can be quite late, so they can be quite lethal, really. So when the protagonists, such as tobacco or excess alcohol, persistently attack normal keratinocytes in your tongue, for instance, you can develop this sort of hyperplasia, mild dysplasia, all the way up to severe dysplasia. And you might get alterations in procancerous elements, and that can then develop into full cancer. And these cancers are also quite metastatic. So all squamous cancers are characterized by a high degree of local invasiveness and a high rate of metastases to the neck and cervical lymph nodes. Invasion of oral squamous cancer cells into maxillary and mandibular bones is also a common problem. So keeping on the theme of metastases, one of the indices that we measure, but I'm not really going to talk about, was anoicus resistance. Anoicus resistance is a prerequisite for metastases. What is it? Anoicus describes normal cells undergo apoptosis when they lose adhesion to the extracellular matrix, a process termed as anoikos from the Greek phrase meaning without a home. Metastatic cancer cells develop the ability to survive and grow detached from the extracellular matrix, and thus are termed anoikos resistance. So the other part of Marilena's thesis, we were looking at that phenotype, and we were able to show that an IL-6 drives anoikos resistance in oral squamous cancer cells. And in particular, we could show that recombinant human IL-6 can drive anoikos resistance in the cells. But for the bioenergetic study then, I was more interested in comparing, contrasting the precancerous dysplastic oral keratinocytes with the cancerous ones. And just to say, the dysplastic ones came from a piece of tongue from a 57-year-old male. The cancerous ones came from the tongue of a 55-year-old male. These dysplastic cells are not cancerous. I mean, the comparisons and contrasting that's been done between the SCC4s and DOCs shows maybe an increased P53, but no changes in RAS proteins and things like that. There's aneuploidy, though, as well, which would be, I suppose, an early index of genetic instability. So when we compared and contrasted the bioenergetics, we saw this. So these are data from a seahorse. The top there shows basal oxygen consumption rates in either DOC cells with the open circle or the cancerous or squamous cancer cells, the black. And there was a difference in cellular oxygen consumption that was accounted for by a difference in mitochondrial oxygen consumption. So I think the first thing to say, though, is, as was alluded to in the previous talk by Dr. Moreno-Sanchez, there's quite significant oxygen consumption in these cancer cells. And the point is there's significantly more in the DOC cells. Is that difference due to a difference in mitochondrial abundance? The answer is no. By measuring citrate synthase, we were able to show, in fact, that there was more mitochondria in the cancer cells than in the DOC cells. So per unit mass of mitochondria, I suppose, these differences are even more, the differences are greater. The extracellular acidification rate, which, again, as our previous speaker alluded to, is an index but not a definitive measure of glycolytic flux, shows that the SCC4 cells have a greater extracellular acidification rate, which may reflect increased glycolysis compared to the DOC cells. So this alone is new information. I mean, I could find one paper in all the literature to measure oxygen consumption in SCC4 cells, produced by a colleague of Eric's and I in Romania, Danina Mutian. She's the only person I know who looked at these oral cancer cells. There was no literature on DOC cells at all. Going deeper then, I wanted to see, like, let's sort of investigate the electron transport system and see if there's any differential activity there. And using the suit protocol, Marilena visited Eric's lab and got this nice result showing essentially that, this is the primary data, this is collated data from several experiments, that the NAD-related metabolism is lower in the SCC4 cells compared to the DOC cells. Again, using a totally different system, just because we borrowed it for a while, the Biolog system, which sort of looks at the oxidation of particular metabolites, again, complemented what we saw in collaboration with Eric, insofar as that we saw, malate flux was higher, or lower, in SCC4 cells compared to DOC. Alpha-ketoglutarate glutarate was lower in SCC4 compared to DOC. It threw up this extra fact here that succinate was higher in DOCs compared to SCC4s. And glutamine metabolism, presumably reflecting glutaminolysis, seemed to be higher in the SCC4 cells compared to the DOC cells. But we also measured the actual activity of complex 1, and interestingly, it was lower in the SCC4s compared to the DOCs. So that's all new information, and that alone was quite interesting. But I said that recombinant human IL-6 promoted anoicus resistance, and the assay time for that is 24 hours. So we just looked at how 24-hour treatment of our cells with recombinant IL-6 would affect bioenergetics. And to our amazement, we saw this. So the DOC cells, no effect of recombinant IL-6, whereas with the cancerous cells, we saw a significant decrease in oxygen consumption. No difference in mitochondrial abundance in that 24 hours, as you might expect, but just to verify there wasn't. And again, the ECAR was raised in the SEC4 cells compared to the DOCs. And again, another bit of information that I should convey here is that we could show in the other study that there were IL-6 receptors in the SEC4 cells, but none in the DOC cells. So obviously this addition of IL-6 complements that observation too. And then finally, the final bit of data then is that just probing again deeper using the Ouroboros and the SWEET protocol, we were able to show that essentially pyruvate-related mitochondrial activity is lower in the cancer cells treated with IL-6. So again, that's news because of all the observations I've seen in the literature, IL-6, if it does anything, it increases oxidative metabolism in muscle, but I've never seen it reported to decrease oxidative metabolism. And this is as far as we've got, and the future work will sort of try and elucidate some of these things. So in conclusion then, a conclusion before this conclusion is clearly the SEC4 cancer cells are less oxidative than the DOC cells, the precancerous cells, but they are to some degree oxidative. And the SEC4 cells seem to have more mitochondria than the DOC cells. The SEC4 cells appear to be more glycolytic if you interpret extracellular flux as that. And then the interesting observation that IL-6 seems to suppress mitochondrial activity and pyruvate may be related activity in the SEC4 cells. And future work will be trying to elucidate how it does that and how metabolism might link to anoicus resistance. So it just remains for me to thank those who did the work and those with whom we got the grant. As I said, Marlena did the work, collaboration with Eric. She was seconded here. This group showed us some of the phenotypic assays, and these were the personnel on the grant that we got. Thank you for your attention, and I'll take questions.
Many thanks, Richie. There's time for several questions from the audience. Yes, please. In the middle.
Yes, any speculation at all what the IL-6 might be doing to depress pyruvate-based metabolism?
I don't. I mean, I could speculate away on covalent modification elements, but again, that's a bit hit and miss in the literature as well for other things that affect complex 1 activity. It might be substrate selection, again, in some breast cancer cell stuff we've seen, and in HeLAs, as mentioned by Dr. Moreno-Sanchez, you can get inactivation of pyruvate dehydrogenase by pyruvate dehydrogenase kinase. So that's something that can be initially easily investigated, but I'm not going to talk about that. We've done some work on that. So it could be substrate supply, or it could be inhibition, for instance, through covalent modification of the electron transport chain.
Other questions?
Thank you very much for the nice slides. Yes. I would like to ask you, so we also made the observation that there is a dose-dependent effect on IL-6 circulating in blood and the correlative nature on impairments in mitochondrial functioning in depressed individuals. And have you done some IL-6 dose-dependent experiments to see whether there is a dose-dependent linearity between IL-6 and the effects on cellular metabolism?
Let me just think about that. I think we have done some dose dependence for the recombinant human IL-6. I'm just trying to recall. This was the optimal concentration for us. That's all I know. Yeah.
And the second part would be, so normally people don't drink regular alcohol because it tastes so good. And if they drink regularly, on a regular base, alcohol, we talk in the psychiatric or psychological field about substance abuse disorders. So my question would be, how strong is an influence of any mental states, comorbidities, substance abuse relevant for observations on a cellular level when it is about cancer progress and treatment responses? Thank you.
That's a very big question that I really can't answer. I mean, there's no doubt that a lot of oral cancers are neglected. I mean, because either dentists aren't trained to pick them up and hospitals don't look for them. So, and yes, people who are drinking methadol or whatever, they're obviously going to be more prone to that damage. But I don't know what to say to that question, really.
Okay, thanks again. No, there's no time anymore. We move on, sorry. Thank you.
First presenter is Lente Lerink, who will talk about the renal respiratory conductance, a complex matter.
Yes, thank you very much for inviting me to this great meeting. Today I would actually like to present some preliminary but exciting findings rather than any conclusions, because we would like to start a discussion about renal respiratory conductance, which turns out to be quite a complex matter. So to begin with, the kidney is actually a really energetic organ and is highly reliant on ATP, and its requirements are actually similar to the heart. And in line with this, if kidneys actually experience metabolic derailment during organ transplant procedures, but also during major surgery, it results in dysfunction, as manifested by delayed graft function or acute kidney injury. So therefore, if we want to limit post-ischemic injury, or prevent it even, we should probably focus on the prevention of metabolic injury or optimal use of residual metabolic capacity. So recently, normothermic machine perfusion, or NMP, has been introduced as a novel method to preserve, assess, and remodel organs ex vivo, and even probably test possible interventions under relatively physiological circumstances. And although this seems to work well for a shorter period of perfusion, there come some challenges for longer periods, especially for the kidney. And these mostly comprise metabolic challenges. So for example, there's excessive lactate production, which is actually indicative for failing renal metabolism. And this implies that basic physiologic metabolic requirements for the kidney are not met during current NMP procedures, let alone the requirements to test metabolic interventions. So therefore, it's actually required to have better tailored strategies for the kidney on NMP. And one of these things may be to have an optimal substrate composition to drive ATP synthesis during normothermic machine perfusion. But what would the kidney actually prefer to make ATP from? So recently, a group from the US introduced the creatine kinase clamp, or the CK clamp, which they use to measure respiratory conductance, which is a measure for the mitochondrial metabolic competence in the presence of the supplied substrates. And for this, they exposed murine mitochondria to a change in ATP-ADP ratio, actually an increase in ATP-ADP ratio by titration of phosphocreatine. And then they measured the responses by all different tissues. And most remarkably, the kidney was one of the tissues that showed a really peculiar phenotype by only responding to the CK clamp in the presence of rotenone and succinate. So we were interested by these findings, but we wanted to validate this in porcine kidneys as well, because we know from previous experiment that there are quite some interspecies differences in metabolic function and as well as susceptibility. So we actually performed similar experiments with kidneys from abattoir pigs. We used permeabilized cortex tissue, which we exposed to the CK clamp, and we measured maximal respiration in the presence of the lowest ATP-ADP ratio, and we also measured the respiratory conductance. And in line with the very sparsely available preclinical data, we also saw that these porcine kidneys did not respond to the CK clamp in the presence of any of these substrate combinations. However, when we added rotenone and succinate, we did see a response. So this is quite interesting. Why does the kidney seem to prefer this, this combination of rotenone and succinate, and thus complex 2-driven ATP synthesis rather than complex 1-driven ATP synthesis? Moreover, the respiration rates were also a lot higher in these conditions. And we don't know. There are actually more questions than answers. So one of the things is maybe, is this not a physiological situation? Does the kidney even respond to changes in ATP-ADP ratio? Or does it have a very constant working rate, and does it therefore should not even respond to such a thing? Is another process involved? We simply don't know. So we need your input. So to conclude, the CK clamp may help to define optimal perfusate compositions to stimulate renal metabolism during pneumothermic machine perfusion. And previous findings regarding the need for complex 1 inhibition to observe a response to a different ATP-ADP ratio in kidneys have been validated but are still poorly understood. Thank you.
Thank you very much. Unfortunately, we don't have time for a question, and I would like to introduce our last speaker, Andras Meszaros from the Medical University of Innsbruck, and he will talk about the effect of static cold storage and machine perfusion on mitochondrial respiration in the liver.
Thank you very much. Mike Murphy once had a talk at a surgical conference, and the title was "What Surgeons Should Know About Mitochondria." So then maybe my title could be "Why Surgeons Should Measure Mitochondrial Respiration." Innsbruck is the biggest liver transplantation center of Austria, and liver transplantation is the only curative method of end-stage liver disease. And many patients, up to 20%, are dying on the waiting list because they don't get the organ. So we should extend the donor pool to help those, but we are living in a society which is getting older, getting obese. Many of these patients are on the ICU, and many have a steatotic liver. And all these organs, or many of these organs, have an increased risk of ischemia, reperfusion, injury, graft dysfunction, and that's the worst, even inferior graft survival. But some of these organs are good. We just need to find out which. And there is a lack of an objective method to find out which are the good organs. We all know that liver has detoxifying and biosynthetic capacity, and to cover this, you need a lot of energy, which are covered mainly by the hepatocyte mitochondria. And the dysfunction of the mitochondria can, of course, be a cause, but also a consequence of injuries, acute or chronic. So if you put this all together, then the pre-transplant mitochondrial respiration could be such a prognostic biomarker. That's how usually a liver transplantation goes, right? So we have a donor, then the graft is taken, it's stored in an ice box, that's the so-called cold ischemia, static cold storage, and we just heard about machine perfusion. Many of the livers will be machine perfused normothermically, then get transplanted, and then we see the clinical outcome. So for both of these cases, only static cold storage, but also with machine perfusion, we had a study. First, we had the livers, we took a biopsy by the end of the static cold storage, measured the mitochondrial respiration and correlated with the clinical outcome. And in the second study, we also measured the respiration by the end of cold storage, but also during machine perfusion, and we correlated with the clinical outcome. So here is the first study. About 43 livers were included during static cold storage, and we aimed at a rapid sample preparation, so a biopsy was taken and processed immediately with the shredder tool of the O2K, and we measured the respiration in homogenates. Here you see the pathway control, NADH, fatty acid oxidation, succinate, mass-specific, and also the FCR of the human liver. You see that NADH contributes to about 10%, fatty acid oxidation 25%, and succinate alone up to 100% of the respiration. Once we stratified the cohort in two groups, livers with a good function and livers with a inferior or worse function, after transplantation, you see that the PL control efficiency, the ATP production efficiency, is significantly higher in those livers which are performing well right after transplantation. In this conditional density plot, you also see that the low PL control efficiency goes with a high probability of early allograft dysfunction and vice versa. In the second study, we included 50 livers undergoing also normal thermic machine perfusion, and 35 of these were then also transplanted. We took the biopsies after static cold storage and also during machine perfusion. Basically, you see that the pathway contribution and the flux control ratios do not change during machine perfusion, and these are the time points. And also the PL control efficiency or the cytochrome C control efficiency remains stable over time. But there is a huge variance of the single liver data here, and that's the question, whether this correlates with the outcome. So these are the correlation analysis, a multiple regression model, and what you see here, that the PL control efficiency, the cytochrome C control efficiency, and the leak respiration correlates very nicely with our clinical endpoints if you measure it during the first 6 hours of machine perfusion. Here are some exemplary data. That's a well-performing liver, high and good coupling, low cytochrome C effect, low leak, and that's a bad one with, of course, different data here. So we characterize the mitochondrial respiration during static cold storage and also during machine perfusion in the liver. And we see that in both settings, the mitochondrial respiration has a predictive capacity for the clinical outcome. So this might be a clinical biomarker. So to answer the question, why should surgeons measure mitochondrial respiration? Simply to save more lives. Thank you very much.
Thank you very much. Unfortunately, we don't have time for questions, and I would like to close the session. So thank you for your attention and enjoy your lunch.